The Neon NxT system streamlines the electroporation process for fast and efficient delivery via simple, flexible, and customizable protocols using our electroporation tip technology, where the electroporation takes place inside of the pipette tip, featured in over 11,000 peer-reviewed publications. The system allows researchers to optimize their electroporation parameters to efficiently transfect hard-to-work-with cells, but also includes over 350 pre-optimized protocols in over 150 different cell lines. Successful electroporation relies on a delicate balance of multiple factors, and optimization of these factors is important for successful electroporation of your cells. Optimizing the pulse voltage, the pulse width, the pulse number, as well as cell density, cell health, along with considerations for payload characteristics, including payload size, type, concentrations, and ratios, is really essential. Another important consideration here is the electroporation buffer used during the electroporation process. Optimizing all of these parameters, we can open new doors for advancements in electroporation efficiency. Now, looking specifically at the electroporation profile, the electric field strength is dictated by the voltage and the width of the electric pulse. Multiple pulses can also be used as a strategy to improve efficiency, viability, and control of the process, making it more versatile and an effective technique for introducing molecules into a variety of different cell types. But aside from optimizing the electroporation profile and the electric pulse, selecting the right electroporation buffers for your specific applications is also necessary. The Neon NxT kit contains one electrolytic buffer, which can be used for all applications, and 3 different resuspension buffers you can choose from to optimize for different applications. The R buffer is optimized for efficient and gentle electroporation of mammalian cells and is really ideal for transfecting a wide range of different cell types, including primary cells and stem cells. T buffer is designed for hard-to-transfect cell types, specifically those that require higher voltages. And finally, the Neon NxT Electroporation Kit also has a genome editing buffer, or GE buffer, which allows you to enhance your genome editing efficiency, especially knock-in efficiencies, where we've observed this in several different cell types, including activated T cells. Now, it's really important to note how electroporation parameters affect your transfection efficiency and cell viability. Right? The voltage applied during the electroporation process determines the strength of the electric field and the extent of the membrane disruption. Higher voltages can cause irreversible membrane damage, while lower voltages may not produce enough disruption to allow for efficient uptake of your payload into the cell. The pulse width is also another important consideration, where the pulse width is the duration of the electric pulse, and it can affect the efficiency of the electroporation. Longer pulses can produce more efficient uptake of your payload, but can also cause more cell damage, while shorter pulses may be less efficient and cause less damage. Finally, the pulse number is also important. This is the number of electrical pulses applied during the electroporation process and affects the amount of material that can be introduced into the cells. Multiple pulses can increase the uptake efficiency, but too many pulses or prolonged pulsing can cause significant cell damage and cell death. Now, figuring out where to start your optimization can be a daunting task, but this can be made a little simpler by utilizing tools like our Neon NxT 24-well optimization protocol. This is a pre-programmed optimization protocol where 24 conditions with various voltages, pulse width, and pulse number combinations are preloaded onto the instrument, both as individual protocols and in a plate format. This provides a great starting point to optimize electroporation for any new cell type, where you can start to establish the correlation between your voltage, pulse width, pulse number, and the efficiency, transfection efficiency of your particular payload into your particular cell type. Now, the results of the 24-well optimization protocol can help you choose a suitable protocol specifically that balances efficiency as well as recovery of your cells. This graph shows the relationship between electroporation knockout efficiencies versus cell recovery and total viable edited cells in hematopoietic stem cells, or HSCs. As you can see, with higher knockout efficiency, cell viability and recovery decrease. So one can choose between the different programs that result in either a higher percent of transfected cells or a higher total number of edited cells, depending on your application and your needs. As a note here, higher viability indicates healthier cells that are more likely to exhibit normal physiological function. And even though initial recovery may be lower, the cells exhibit a robust ability to recover over time, reaching similar viabilities in a short amount of time. Aside from the 24-well optimization protocol, you can also use Transfection Lab, which is a digital experiment design application that helps you design your own electroporation experiment in different formats. After setting up the parameters of your experiment, Transfection Lab automatically generates a step-by-step experimental guide tailored for your experiment. Now, also, saved protocols and plate formats on the Transfection Lab can also be accessible and can be directly loaded onto our Neon NxT instrument utilizing our Instrument Connect features. You can learn a little bit more about Transfection Lab by watching the video below here as well. Other important considerations, including downstream optimization, such as seeding densities, recovery, and cell culture media, as well as feeding schedules, are really important to consider. And not to mention that the inclusion of controls during your electroporation experiments is critical for benchmarking and troubleshooting efforts. And to learn more about our Neon NxT Transfection Lab and view more resources, please go to thermofisher.com/neonnext.